Hello student, I am your educator Miss Vidisha Kapse from Department of Zoology, SVKT Art, Science and Commerce College, Deolali Camp, Nasik. Today's lecture is for FYBSc students of NEP 2020 pattern. The subject I have chosen is the cell biology and the topic is plasma membrane and its transport system. Let us start our lecture. First, we are going to study the introduction of the plasma membrane. As we know, plasma membrane is the important cell organelle which covers the all cell, which covers the membrane of the cell. So, it is also called as cell membrane. It also delimits the cytoplasm and also covers the all cell organelles. So, let us see its definition. The plasma membrane is the outermost boundary of the cell separating cell's internal environment from the external environment, and it plays significant role as it acts as a selective barrier which controls the movement of substances in and out of the cell, hence helping maintain the homeostasis. Homeostasis means the balance between internal and external environment of the cell. So, plasma membrane allows only selected molecules to pass through it. Thus, it helps in maintaining the homeostasis. Then, the location of it found in all living cells from simple unicellular prokaryotes to the multicellular and complex bodied eukaryotes. Okay, now discuss about the structure of plasma membrane. The plasma membrane is made up of different constituents. In this, one of the main constituent is the phospholipid bilayer, which forms the basic structure and give the proper shape to the plasma membrane. The phospholipid bilayer means two layers of phospholipid that have the two faces. One is the hydrophobic tail that faces the inward of the plasma membrane. Hydrophobic means which hates the water, hence this layer avoids the water. After that, second face is the hydrophilic head that faces the outward of the plasma membrane. Hydrophilic means which makes the bond with the water. So, this layer faces the water, that means the outer membrane of the plasma membrane. After that, second constituent is the proteins. The proteins are of different type. One is the integral proteins and second is the peripheral protein. The integral proteins are also called as transmembrane protein and peripheral proteins are also called as exterior proteins. The third constituent which plays the important role is called as cholesterol. Cholesterol is important because it helps in maintain the membrane fluidity. That means it gives the cell elasticity so that cell can change its shape according to the function. After that, next is a carbohydrate. Carbohydrates are involved in cell recognition. That means it gives the cell its specific identity so that the specific proteins or the hormones can enter to the specific cell. So, let us discuss about the structure and function of the phospholipid bilayer. As we discussed earlier, it has a two layer in which the phospholipid has the two structure. One is the one head and there is a two tails. The head, which is the polar or the hydrophilic, that faces the outward of the layer, which makes the bond with the water. The second one is the hydrophobic tail. There are two tails present which are non-polar, that makes the inner side of the plasma membrane, which faces away from the water. Then phospholipid bilayer performs the important function as it creates a selective barrier, means only allowed the selected molecule to pass through it. Second important function is it maintains the membrane's integrity and fluidity. Next is the membrane proteins. So, there are two important membrane proteins are present. One is the integral protein that embedded in the membrane and acts as a channel or the transporter. The integral proteins transpasses the cell membrane and it important in the transportation system. Second one is the peripheral protein that are attached to the outer and inner surface of the membrane and they are involved in the signaling and structural supports. That means when specific signal have to pass to the cell, that time these signals signals are transferred by the peripheral proteins, and the changes occur inside the cell, and a response is given to the outside signaling. After that, there are different types of membrane proteins are classified according to their functions. First is a channel protein. This allows the specific ions or molecules to pass through it. When there is transfer of molecules occur, that time channels get open. After when there is no function, that time channels remains closed condition. After that, there are some carrier proteins also present that binds to the molecule and helps them to cross the membrane. Third one is the receptor proteins. Receptor proteins are specific to specific its ligands. That ligands are sometimes the external signals which binds to receptor, and these ligands are carried out inside the cell. For example, hormones. After that, fourth protein is the enzymatic proteins that facilitate chemical reaction on the cell surface. This is a fluid mosaic model. There are different models are proposed by the different scientists to give the proper structure of plasma membrane, but one of the accepted model is the fluid mosaic model. The model says that it describes the plasma membrane's dynamic and flexible nature, where lipids and proteins move within the layer like a fluid. That means plasma membrane allowed the free movement of lipids and proteins from upward to downward direction, from lateral side, also flip-flop movement. So this gives the fluidity to that membrane. This well-accepted model is proposed by the scientists S.J. Singer and G.L. Nicholson in 1972. There are two words. One is the mosaic and second one is the fluid. The mosaic means the membrane is the patchwork of different molecules. Means that the surface of the membrane is not a plane. It contains different constituents like phospholipid, proteins and cholesterol, which give the mosaicity to the plasma membrane. Second one is the fluid. Fluid refers to the membrane's flexibility and its ability to allow the lipid and protein movement within the bilayer. That means it gives cells the elasticity, and cell can change its shape according to its function. This is a diagram of fluid mosaic model. Here we can see that there are two layers of phospholipid present, in which the integral membranes are present. Integral membranes transpasses the cell membrane, and it is modified into channel proteins and alpha helix proteins. This both integral proteins helps in transportation of the molecule. Then there are some attached peripheral proteins are also present. These are attached to outer and inner layer of the plasma membrane. Okay. Peripheral proteins are also called as exterior proteins. These are modified into globular proteins. Also, there are next constituent present, which is called as carbohydrates. Carbohydrate provides the identity or tag to that cell. So, carbohydrate are associated with the different molecules. When it is associated with the proteins, then that time it is called as glycoproteins. When it is associated with the lipids, then they are called as a glycolipids. Then there is another which is very important molecule is present, which is known as cholesterol. Cholesterol plays important role in fluidity of the plasma membrane. So this is a fluid mosaic model. Now let us discuss about the function of plasma membranes. There are very important functions are performed by this cell membrane. One is the selective permeability. As we discussed earlier, it controls what should enter and exit the cell. For example, when there is deficiency of nutrients occurs in the cell, that time nutrients are carried out from outside to inside the cell. When there is the need of to throw out the wastage from the cell, that time wastage are removed from the cell, from inside to outside the layer. Then second function is the protection and support. It acts as a physical barrier against harmful substances and pathogen that causes the diseases. Then next function is communication. Cell membrane receptor transmit signals between the cell and its environment. So, cell to cell communication is carried out by the plasma membrane, and also there are some signaling cascades are also performed in the cell. The next function is cell recognition. As we know, carbohydrate perform role in the cell recognition. The carbohydrates associated with the glycoproteins or the glycolipids allows cells to recognize and interact with one another and giving its identity. So next part is the very important part, that is the transport across the plasma membrane. As we know, plasma membrane plays important role in transportation, so cell can perform its various functions. The plasma membrane is selectively permeable, meaning only allowed some substances to pass more easily than others. So, the transport system is classified into two types. One is the passive transport and second is the active transport. The passive transport does not require energy because the substances or the solutes move along the concentration gradient. That means the transportation of substances occur from high concentration to its low concentration. So, no energy is required. But in active transportation, there is requirement of energy occur in the form of ATP because substances have to transport against their concentration gradient. That means the substances have to move from low concentration to high concentration. So, adenosine triphosphate provides the energy for this process. Okay, this is a flow chart of membrane transportation. There are two types of molecules have to transport inside the membrane. One is the small molecule and second is the larger molecule. The smaller molecule transport by the two systems. One is the passive transport system and second is the active transport system. The passive transport system is classified into two types. One is the simple diffusion and second is the facilitated diffusion. In the simple diffusion, the transportation of water, non-polar substances and ions occurs. In facilitated diffusion, there is a requirement of channels and permeases occurs. Then second type, which is the active transport, which requires the energy, there is the requirement of pumps and co-transporters occurs. Then for the larger molecules, transportation of the larger molecules, there are three systems occurs. One is the endocytosis, second is the exocytosis and third is the pinocytosis. Let us discuss in detail about that. Passive transport. Passive transport doesn't require energy and it is classified into different types like a simple diffusion, facilitated diffusion and third one is the osmosis. In the simple diffusion, the movement of molecules occurs from an area of high concentration to an area of low concentration. That means it occurs along the concentration gradient. For example, in the cell, there is deficiency of oxygen occur. That time, cell takes the oxygen from the outside of the cell. When there is the concentration of CO2 increases inside the cell, that time excess carbon dioxide is expelled outside the cell. This is called as simple diffusion. Second type is the facilitated diffusion. It is same as the simple diffusion, but it uses the membrane protein that helps molecule that cannot directly pass through the lipid bilayer. For example, glucose and iron. Because of hydrophobicity which is present in the plasma membrane, some molecules require some of the carrier proteins for their transportation. So there are two proteins, channel proteins, which forms pore for the specific molecule, and also carrier proteins which binds and transport the specific molecule inside the cell. The third type is the osmosis. The osmosis is called as special case of diffusion, where water molecules moves across a selectively permeable membrane. Okay, in about two types, we seen that there is the transportation of solute molecules occur, but in osmosis, the transportation of solvent molecule occurs. This is the difference between two processes. So here solvent, that means the water molecules, where the movement occur from low solute concentration to high solute concentration. That means from high solvent concentration to low solvent concentration. This is called as osmosis. So this is a diagram which is showing difference between simple diffusion versus facilitated diffusion. We can see that in the diagram, in the first simple diffusion, the diffusing particle easily transferring from one layer to another layer. That means it is easily transported through the plasma membrane because from its high concentration to its low concentration. But in facilitated diffusion, there we can see that the diffusing molecules require some carrier protein to pass the membrane. So carrier protein helps here to pass the molecules from their high concentration to their low concentration level. This diagram showing the osmotic gradient. Osmosis means that the solvent, that is water, moves from lower to higher solute concentration. In the first jar, we can see that there is a partition created by the semi-permeable membrane. The yellow dots showing the solute particles. So we can see that in the one side there is a low solute concentration seen, and in another side high solute concentration is occur. So at that time the water start moving from low concentration to high concentration. In the second jar, we can see that this is a final state in equal concentration. That means both layer has the equal concentration of solution, solute plus solvent concentration. So, but by this process, there is some difference occur which creates the osmotic pressure. This osmotic pressure is important because it helps in maintains shell shape and functions. Okay, so this is a we earlier seen that this is a movement of water from low solute concentration to high solute concentration, and there is the importance of osmotic pressure also occurs. So here we can study the three conditions. One is the hypotonic solution condition, second is the hypertonic osmosis, and third one is the isotonic osmosis. In the hypotonic solutions means that it has low solute concentration and high solvent concentration. When we place our cell in that time the outside water start entering inside the cell. That time our cells get swollen out. In the second condition, in hypertonic solution, that means which have high solute concentration and low solvent concentration, at that time water start leaving the cell. So cell get shrink out. In the third condition, in isotonic solution, which have same solute and solvent concentration as that of cell, when we place cell inside that, there is a no net water movement occurs. Okay, this diagram showing three solutions what affect the cell. Here we chosen the two cells, one is the animal cell and plant cell. How the solution affect our animal and plant cell. In the first condition, in hypotonic solution, the water start entering inside the cell. So that condition we are called as it is lysed animal cell. Also in same for the plant cell, when water enters inside the cell, that time we call it as a turgid plant cell. For isotonic solution, normal animal cell and flaccid plant cell is same. In hypertonic solution, cells start leaving the water. So this condition is called as shriveled animal cell. For the same for the plant cell, this condition is called as plasmolysed plant cell. Okay, next move on to the next point, which is a active transport. As we know, it occurs against their concentration gradient, so ATP is required as a form of energy. So this we study by the very famous example, that is a sodium potassium pump. The sodium potassium pump transports 3 sodium ions out of the cell and 2 potassium ion inside the cell. Which this is important because it is helps maintain the proper ions balance and resting potential of the cell. Okay, the sodium potassium pump, the cell requires the high intracellular potassium concentration and low sodium concentration because it is needed for survival of the cell and also required for to do the different functions. The high cellular potassium is required for the optimum glycolysis. Glycolysis means the breaking down of complex sugar into simple sugar so that cell can use it. Okay, for this process, the pyruvate kinase is one of the important enzyme which is required, which depend upon the potassium concentration. Also, potassium is required for protein biosynthesis. So, the sodium and potassium gradient is also needed for the transmission of nerve impulses. So, the message can be transferred to the brain and our body can respond to the external stimuli. This pump is responsible for maintenance of high potassium concentration and low sodium concentration inside the cell. This process is carried out by the enzyme called as sodium potassium ATPase. This helps in pumping out 3 sodium ions outside, pumping out the 3 sodium ion outside the cell and taking the 2 potassium ion inside the cell. So this reaction showing the what happens in this condition. 3 sodium which is present in the first reaction is occurring inside the cell. There is a 3 sodium ions and 2 potassium ions which are present outside the cell. For this process, the ATP is required. In the second condition, 3 sodium ions are expelled out and 2 potassium ions are taken inside, and the hydrolysis of the ATP molecule occurs, which is converted into ADP. Okay, this diagram is showing the sodium potassium pump, how the ions move from inside and outside of the cell. In the first condition, we can see that there is a lot of sodium ions are present inside the cell. When ATP molecule get attached to that pump, that time the carrier molecule changes its shapes and 3 sodium ions are expelled out. Then ATP is hydrolyzed into ADP. Also there is carrier molecule change its shapes and 2 potassium ions are taken inside the cell. This diagram is showing how it works. Okay, now move on to the transportation of the larger molecule, which contain endocytosis and exocytosis. Now let us discuss about the endocytosis. Endocytosis means taking the molecule inside the cell, that is taking the substances into the cell by forming the vesicles. This process involves 2 types. One is the phagocytosis. Phagocytosis means transportation of the solid particles and pinocytosis means transportation of the liquid particles. Okay, so phagocytosis is also called as cell eating. That means cell engulfs the larger particles and giving rise to an internal compartment called the phagosomes. In the pinocytosis, this process is also called as cell drinking, which the cell ingests the extracellular fluid and its dissolved particle. This is the process of endocytosis. Here we can see that the larger particle, when come in contact with the plasma membrane, that time plasma membrane start invaginate and covers that larger particle. After covering that particle, there is the pinching of plasma membrane occur and vacuoles are taken inside the cell. After that, there are some enzymes are present inside the cell that dissolves these vacuoles and these larger particles are taken inside the cell. Same occurs in the pinocytosis. Here the liquid particles, when come in contact with the plasma membrane, same procedure happens and the the liquid molecules taken inside the cell in the form of vesicles. There is also another modified type which is called as receptor mediated endocytosis. So the specific ligands which have the specific receptor, they ligands get attached to the receptor. That time conformational changes occurs and membranes start invaginate and coated vesicles are formed, and these particles are taken inside the cell. Okay, next process is the exocytosis. Exo means outside and cytosis means cell. So this is the process of expelling the substances from the cell via vesicle fusing with the plasma membrane. Okay, there is a release of micromolecules from the cell to outside occurs with the help of the Golgi apparatus. Golgi apparatus plays important role in the cell's transportation. So in this diagram we can see that the vesicle which are pinched off from the Golgi apparatus start migrating towards the plasma membrane. After coming in contact with the plasma membrane, it start dissolving, and the substances are expelled out to the extracellular membrane. Here we can see, we can study the example of insulin. Insulin is transported by this exocytosis process. This is the difference showing the transportation between passive transportation and the active transportation. So in passive transportation, no energy required and particle move from high to lower concentration. But in active transportation, energy is required and solute moves from its low concentration to high concentration. So there is a type present in the passive transportation like simple diffusion, facilitated diffusion and osmosis. Here we studied the sodium potassium pump, proton pump, endocytosis and exocytosis. There are some points which are similar in both, like both are involved in movement of molecules along the concentration gradient, and also it is important to the cell functions. Role of membrane protein in the transportation. We studied it earlier, but now discuss it in detail. The transport proteins which are help in facilitated diffusion and also in active transportation. First is the channel proteins. These forms the pores only allowed the specific molecule to pass through it. Example we can see that here, the ion channels or the aquaporins. Aquaporins are the channel which are specific for the water. When water have to taken inside, that time these channels gets opens and allow water molecules to pass through it. Whenever there is no requirement of water, that time it remains in closed condition. Second is the carrier proteins, which binds to specific molecule and change its own shape and helps the solute to cross the membrane. Third one is the receptor protein, which receives the signal and initiate response inside the cell and substances taken inside the cell. This is the diagram of transport proteins. First is the channel proteins. Channel proteins start when they get open, which allows the molecule ions to pass through it. But in carrier proteins, they have to change their shape. They first attach to molecule and change its own shape and transport the molecule inside the cell. There are another transport system occurs like a uniport system, symport system and antiport system. In the uniport system, only a single molecule is taken inside the cell or expelled outside the cell. It involves the movement of only single molecule. For example, transport of glucose to the erythrocytes. Then second condition is the symport system. Symport system means there is a simultaneous transport of two molecules but in the same direction. For example, sodium and glucose pump, which expelled or taken inside the sodium and glucose at a time simultaneously. But in antiport system, third type is antiport system, in which the simultaneous transport of two molecules occurs but in opposite direction. That means one molecule are expelled out, simultaneously second molecules are taken inside. For example, exchange of calcium exchange of chlorine and HCO3 in the erythrocytes. This is the summary we have studied today. Today's in lecture is the plasma membrane is selectively permeable, which controls entry and exit of substances via different transport system. Also, passive transport does not require energy, while active transport requires energy in the form of ATP. The fluid mosaic model, which describes the structure of plasma membrane and the dynamic interaction of its component. And membrane protein plays crucial role in transport and cell communication. This is all about today's lecture. Thank you.